DNA replication is the process that happens during S phase of the cell cycle. DNA replication is a faithful copying of the double-stranded DNA sequence along the length of a chromosome. Cells divide into two new cells by cell division, and the primary mode of cell division is mitosis, which is involved in growth, cell reproduction, and cell replacement. Reproduction requires mitosis for asexual reproduction and meiosis for sexual reproduction. Regardless of the type of cell division, the DNA has to be faithfully replicated so that two new cells, or four at the end of meiosis, have the DNA necessary for the cell to function. Remember that DNA provides the instructions for the production of proteins required for cell function. So, all cells need all of the DNA, and the DNA must be copied before cell division. One cell dividing into two new cells during mitosis is providing new cells for a growing organism, or maybe new cells in order to repair wounds. You start out as a single cell, and by the time you're trillions of cells, it's based on and due to the fact that that one cell initially divided into two. So all of the cells in your body have all of these sets of instructions for the production of every possible necessary protein. Now, we're going to see that it turns out in cells, not all cells are actually going to make every protein that's encoded, but every cell has the instructions for making every protein. The process of DNA replication involves this original double-stranded DNA molecule separating into two template strands. These are called the parental strands, and they serve as templates for the incorporation of new nucleotides based on base pairing rules so that the two new molecules of DNA are identical to the original, and the two molecules consist of one parent DNA and one daughter molecule, again held together by hydrogen bonding between bases. Ultimately, these two molecules are going to be separated from each other during cell division, and one molecule will be in one cell, the other in the other cell, but those two new cells will have the exact same DNA content as the original cell that divided into two. It's actually probably more appropriate to look at DNA replication like this, where we have our double-stranded helical chromosome, and it is separated into what's called a replication fork. Each one of these strands is serving as a template for the single nucleotide incorporation of the new daughter molecule. And it's a step-by-step process. Each nucleotide is added one at a time. But you can see then that these two molecules are going to consist of both parent DNA and new daughter DNA. Along the length of a double-stranded DNA chromosome, there are origins of replication, and this is the place where replication is going to start. At the origin, the DNA is separated by proteins that are called initiator proteins. And these initiator proteins pull apart the initial double-stranded DNA region to create the two single strands. In gray is the daughter DNA, and the daughter DNA is synthesized in a bidirectional manner from the origin of replication, and it's happening on both strands. So as this replication bubble opens, the two forks are extended until finally the replication bubbles meet each other to create our two new daughter DNA molecules consisting of one parent and one daughter. These two remain connected to each other until M phase, until cell division. And remember, these two connected to each other are the sister chromatids, those sister chromatids that get separated during cell division. Now, I also want to point out one important feature of double-stranded DNA. During my discussion of nucleic acids early on, I mentioned that there was a 5' end with a free phosphate and a 3' end with a free hydroxyl. And these two ends are distinct from each other. These are like a north and a south pole, a 5' pole and a 3' pole. The other strand of DNA is an opposite orientation to the original. So notice that the 5' ends are in opposite orientation and the 3' ends are in opposite orientation. When DNA unwinds, now we're looking at just one replication fork, new DNA is going to be made always in its 5' to 3' direction, and it's going to be made anti-parallel to the template strand. Anti-parallel is how we describe these two strands running in opposite directions. The molecule that creates new DNA is called DNA polymerase. And DNA polymerase reads a template strand and base pairs with the template, always in a growing 5' to 3' direction. And it has to be going in this direction, 5' to 3', and it has to be making DNA anti-parallel to the original template. Now on this strand, notice that the template on the right here starts at the 5' end because these blue strands were initially anti-parallel to each other. In order for DNA polymerase to satisfy that requirement of synthesizing 5 to 3 and anti-parallel, the fragments on this strand have to be synthesized discontinuously. So this molecule was made when the fork had opened up just a little bit. Then, as the fork opens toward the end of the chromosome, a new fragment is initiated, and DNA polymerase is always making new DNA in a 5' to 3' direction, anti-parallel to the template. This synthesis at the top is continuous. We call that strand the leading strand because it moves a little bit quicker. This strand is called the lagging strand, and it's synthesized discontinuously or in pieces. You might see in sources that these pieces are called Okazaki fragments. Okazaki is just the name of the man who described the synthesis on the lagging strand. But ultimately we want this strand to be continuous DNA, and so what happens is this little enzyme down here called ligase comes in after fragments have been made and seals a gap in between these fragments. That's because DNA polymerase is always adding to the molecule behind it. So when DNA polymerase bumps into this first fragment that it made, it can't seal this little fragment. Ligase is the molecule that comes in and seals the fragment. And notice that overall, DNA replication is in this direction because this fragment was made first, then this one, and all of the synthesis ultimately follows the opening of the replication fork. So, if we look at an entire bubble, we have continuous synthesis and discontinuous synthesis, continuous synthesis and discontinuous synthesis. On both templates, there is a continuous and discontinuous synthesis going on, but ultimately ligase will seal so that there's no indication of where lagging or leading strand synthesis occurred. And these molecules will be anti-parallel with correct 5' and 3' orientations. After replication, each chromosome has been duplicated, and the duplicated chromosomes are connected to each other and called sister chromatids. They should be identical to each other because remember, this is based on base pairing. Now mutations or changes in nucleotide sequence can occur. DNA polymerase makes mistakes. But DNA polymerase can fix some of its own mistakes, and we have repair mechanisms in the cell that come in and fix mismatched base pairs to the best of their ability so that the DNA is faithfully replicated. Now when cell division occurs, cell division will allow the distribution of these two chromosomes to each new cell. The sister chromatids, when they separate, are now considered to be their own chromosome. But initially, two sister chromatids connected to each other are considered one chromosome. And the reason why they're considered one is because of the fact that they're identical and they say the exact same thing. The instructions for making proteins are identical.